Hi, my name is Aaron Houston, and this is a presentation on cell theory, including the structure of different types of cells, prokaryotic and eukaryotic, as well as some different causes of diseases such as bacteria, viruses, and prions. So before we actually get into the detail of the materials, I just want to talk about some breaking news that happened this week. So this is February 2017, and earlier this week, NASA actually announced that they've discovered 7 Earth-like planets around a star. The star is 40 light years away. It's a dwarf-type star. It's called TRAPPIST-1. Now, why this is important is because these 7 Earth-like planets are actually in the habitable zone around the star, and they're only 40 light years away. So this is a pretty close planetary system, and it's the first time we found so many Earth-like planets around a single star. So looking for life beyond our solar system, this is a very, very likely place that we're going to find it. So it's very exciting news. The reason I bring this up is because this presentation is all about life. So finding life or possible places for life in other parts of the universe, let's start talking about what life is. In order for something to be considered alive, scientists generally agree that it has to have 8 common characteristics. All right? It has to have 8 things that all life that we've found has in common. So the 8 things that all life has, the first one, all life maintains order inside themselves. So inside they are made of certain parts, and normal things will break down over time, but life will actually start building up their parts over time as they break down. So if something gets damaged, something that is alive will actually rebuild it, maintain it, try and keep it organized inside there instead of let it stay that way. You can see this when an organism, such as a person, gets a cut or gets damaged. Their body will actually fix itself. They will try to rebuild what was destroyed, unless they're no longer alive, in which case they'd be dead, and then the cut stays there forever. Living things do this, non-living things do not. Second thing that all life has in common, they regulate their systems. This means that they purposely try to keep things the way they are. They try to keep them at equilibrium. For example, temperature. All living things try to maintain at least some semblance of temperature, at least a lot of them do, animals do, because an ideal temperature is how they keep things going, keep the reactions they need to stay alive. Number 3, all living things respond to their environment in some fashion. Animals will respond to their environment, like if they hear a loud noise, they'll respond to it, they'll see it, they'll look at it. Plants do the exact same thing, except they don't really respond to noises or other stimulus that animals would, but they do respond to things like water and light. They will actually turn leaves towards the light to actually try and get more energy out of that light. Number 4, all living things will transfer energy around itself. This means that sometimes the energy is created or digested in one part of the body, and then that energy is transferred to other parts that need it. But they'll also need to transfer energy to and from the environment. Living things actually need to get energy from their environment. Animals need to eat things. Plants need to absorb sunlight in order to continue existing. They need to get energy from their environment, and they put energy back into their environment as well. They radiate heat. They give energy to other organisms if they get eaten. So it's transferring back and forth. Number 5, all living things grow and develop. They'll start off small, and they'll get bigger, and eventually they'll develop different parts, but they will actually grow and develop. And then eventually, all living things will also reproduce. They will have offspring. Okay? People have children. Plants have seeds. Bacteria will divide and create more bacteria. All living things reproduce. It's a common characteristic. Number 7, because they reproduce, all living things will actually evolve over time. Now, evolution does not happen with a single organism. This is because all living things reproduce. So, as you go from parent to offspring to the next generation after that, small changes start developing. And so over generation after generation, those small changes start to make into significant changes. And several generations down the road, you have slightly different organisms. Eventually, over many, many, many generations, you might even have a new organism, something that looks completely different. But it's a very slow progression from organism to offspring, from parent to offspring. And last but not least, and this is one most important that took a very long time to discover, all living things are made of cells. Those cells, which of course contain DNA, right, genetic material for passing those things on. Or RNA. Some living things will actually contain RNA. Now, I'm not saying that all life is similar, but I am saying that all life has at least these 8 things in common. Right? It maintains order, regulates our system, responds to the environment, transfers energy to itself to and from the environment, it grows, it develops, it reproduces, it evolves over time, and it is made of cells which contain DNA. Anything that is not alive does not have all 8 of these characteristics. It might have a few of them. For example, fire, which is not considered alive, does actually have several of these characteristics. Fire does respond to its environment. It does transfer energy around. It needs to eat things. It needs fuel. It does grow and develop. All right. However, fire does not evolve over time. It is not made of cells, and doesn't actually try to maintain order or regulate its systems, which is why fire is not considered a living thing, even though it does have a few of these characteristics. So things that we do consider alive. Plants and animals are the easy ones. Look around, we have plants, we have animals, we know those things are alive. Talking about what they have in common and what they don't in just a minute. But it took a long time for us to get into some of the other more intricate definitions of what are alive. Fungi, which for a long time was considered a type of plant, we now know is not a plant. It's actually more similar to an animal, but those are considered alive. And then it wasn't until we actually discovered bacteria and other microorganisms that we got into just the mechanics of that whole idea of needing a cell, the structure of needing a cell. So how do we actually discover bacteria? It starts off with this guy named Antoine von Leeuwenhoek. I cannot pronounce his name, but I'm trying my best here. He was actually a linen merchant in the 1600s. Now, linen merchants back then, they sold linen. Linen is a type of cloth, and in order to determine the quality of that cloth, they had to look at the thread count of the linens that they were looking at. So they would look very carefully using a magnifying glass at the different threads and how close together they were. And you had to use a magnifying glass because really quality linen would have very, very fine threads that were really close together. So he was actually really skilled at making these magnifying glasses. And because he was so skilled at it, he actually went beyond that. He tried making a microscope. So he invented a new type of microscope, which is actually a diagram right here. It's small, and it's pretty simple, actually. But this very, very careful microscope, you put the specimen right here on this pin, and then you look through the eyepiece, look through the lens. You can use these other knobs to focus it. So you actually had to hold it up to your eye. You just get this tiny little specimen on the pin there. You can actually see microscopic objects. He's the first one to actually see some of these microscopic organisms. So some of the things he might have seen when he looked through this kind of look like this. This is pond water, one of the things that he tried looking at. He also saw blood cells, and he saw sperm, but he would look at these and he'd see these little moving things that kind of look like animals in the pond water. So he named them animalcules, because they were similar animals. He imagined them as tiny little miniature animals living in everything around us. Now, he decided to communicate his findings to this guy named Robert Hooke. Robert Hooke had also tried looking through microscopes and trying to figure out what was going on at the microscopic level. He had a microscope as well, but not as good as the one that Antoine von Leeuwenhoek had. All right, so Robert Hooke had his own microscope, and he had actually done research on cork a few years earlier. So he had used his microscope to look at cork. And he had looked very carefully at cork, and he could just barely see this structure here of cork. And it was actually Robert Hooke who had coined the term cell, because they look like prison cells to him. So he would named this structure here of cell because he could actually see these cells in a cork because they're quite large in corks. But he actually tried to look for these microorganisms in pond water, and he had difficulty finding them. He actually had to modify his microscope before he finally saw them as well. But he is the person who actually coined the term cell. So the big idea from all of this is that the cell here is the basic unit of life. So we're kind of drilling down in there. Robert Hooke came up with the idea that living things can be made out of cells as one of the basic units here. And we have these microorganisms that are really, really small. Now, a little while later, early 1800s, we had these two scientists, Theodor Schwann and Matthias Schlenden. Now, these two did not work together. One of them was studying animals, he was into anatomy, and the other one was studying plants, he was a botanist. Now, both of them were looking at microscopes, using these microscopes to actually study the tissues of their various specimens. And Matthias Schleden, who studied plants, was noticing common things between all the plants. He was noticing similar structures between all of them. Theodor Schwann was also noticing similar structures between the tissues of animals in his anatomy. So if we get up some of the things they were looking at, he was noticing that it didn't matter which animal you looked at, brain tissue always had this exact same structure right here, and muscle tissue had a exact same structure from animals, doesn't matter what the animal was. They always look similar. This is actually brain tissue right here, so we're looking at different nerves right there, but it didn't matter the animal, always had that structure. And even the tissues of muscles and brains, you could see similarities there as well. Matthias Schleden was looking at plants, and he was noticing that they had the same structure from plant to plant. There's always these little rigidly shaped things like this with the little dots in it. And it wasn't until they got together, they actually got together at a dinner and they were discussing their work at this dinner, that they realized that the commonalities between the plants and the commonalities between the animals were the same. All of them, the big idea here is that all of them were made out of the same thing. They're all made out of cells. So at the time, really, it was only plants and animals. We were just discovering microorganisms, but it was these two getting together and realizing that the things they were noticing were the same in the plants, and the things that he was noticing were the same in the animals, were the same between plants and animals as well, meaning that pretty much anything that was alive had to be made of these things. It was a common trait between all living things, and not just common between plants and common between animals. All living things actually had this common trait, this idea of cells. Fast forward a few years, we had Louis Pasteur entered the picture. Okay, now Louis Pasteur is more famous for his pasteurization process, but just to give a little background of what he was trying to investigate here, there was this idea called spontaneous generation. Now, what spontaneous generation is the idea that a living organism will actually spring forth if you have the right ingredients, the right inorganic ingredients. For example, they actually thought that mice would actually come from dirty cloth and grains. So if you had some dirty cloth and some grains and you mix them together and the conditions were just right, it would actually spontaneously generate mice. You'd actually get a little baby mice growing. And there were people who did experiments where they mixed these two together and let them sit for a few weeks, and they found mice in them. So, of course, it was spontaneously generating mice whenever you mix these two ingredients together. Nowadays, we think that sounds crazy, but that's because of what Louis Pasteur actually discovered, what his experiment was. So what he did, at this point, we knew about microorganisms, and the idea was that microorganisms would spontaneously generate wherever we had water, wherever we had nutrients, wherever we had air. So need the right ingredients and we'd get microorganisms. So he designed an experiment to actually disprove that. His experiment was this. He took some water and added some nutrients to it, so he made a broth, and then he boiled that broth to kill off any microorganisms, but he did it in a very special flask. Notice how the lid to this flask is. It's actually got this curve to it. So he would boil it in there, make sure there were no microorganisms inside this nutrient broth. But because of this curve, it's actually still open to the air, all right? but it's unable to actually let in anything except air. Like, you can't get any liquid or dust or anything. They would all the liquid and dust would actually settle in this curve. Only the air was actually able to make it all the way through. So why he did this is because he wanted to prove that you could get all of the ingredients mixed together, air, water, nutrients, everything like that, and nothing would grow. He let this sit there, and because nothing else besides the air could get in, you didn't get any microorganisms. It remained free of it several weeks later. Nothing was growing. But then all he had to do was break it off so that you could actually get dust particles and other things besides air into the flask, and suddenly you got microorganisms growing. So he was actually able to prove that it was not random things mixing together, or the air and the broth and the water mixing together and spontaneously generating these microorganisms. What it was other microorganisms that happened to make their way into the flask, and then once they were there, they were able to actually grow. So you would get microorganisms growing in there. But without the other microorganisms, you couldn't get it. Big idea that he came up with was that all cells, all living things actually had to come from pre-existing living things. So it wasn't until a little while later when scientists actually observed a cell dividing and seeing that it was a cell that divided into two cells. And so we know that all cells actually come from pre-existing cells, but it was this that showed that it had to be a living organism to create more living organisms. So this idea of cell theory, kind of general summary here, really changed the field of biology. Before cell theory, all biology was trying to find living organisms and categorize them and see how they were related and try and name them. It was a system of categorizing living things, but it wasn't understood exactly how living things came to be or what all living things had in common. It was cell theory that actually combined it all. So the basic tenets, quick summary: cell is the basic unit of life. All living things are made of cells, all right? And all cells come from pre-existing cells. Now, there are actually many different types of cells. So there are two basic types: prokaryotic cells and eukaryotic cells. So let me discuss the differences and similarities between these two. Both of these cells consist of a plasma membrane. So I'm going to draw a plasma membrane here. They both have plasma membranes. That's what they have in common. They also both have cell walls, although this is not guaranteed. Prokaryotic cells will definitely have a cell wall. Eukaryotic cells sometimes have a cell wall and sometimes they don't. They do when they're talking about plants or fungi, but they do not if we're talking about animals. So this is an exception to some eukaryotic cells, but all prokaryotic cells have cell walls. Now, both of them have a fluid inside of them, which we call the cytoplasm. This fluid flows around. It's got nutrients, it's got water, it's got all sorts of things floating around it, but they both have this fluid. And they both have things called ribosomes. All right, now ribosomes are tiny little factories that float around the cytoplasm that actually create proteins. So they get instructions from the messenger RNA, and the messenger RNA, whenever it comes up to them, will actually tell them which proteins to make, and then they use the nutrients in the cytoplasm to make whatever proteins that they're instructed to. So both of them have these ribosomes. All right, now here's where the differences come in. The eukaryotic cell actually has a nucleus in its center. So the nucleus is where it actually stores its DNA. So the DNA is stored in nice little packets here in the nucleus, whereas with the prokaryotic cell, there is no nucleus. It doesn't have this contained area for its genetic material. Its DNA or its RNA, some of them just have RNA, float around in the cytoplasm, just kind of float around and use whatever they need. That is the biggest difference between them. DNA is contained in a nucleus, DNA just kind of floats around. The other major difference is that eukaryotic cells will actually have organelles floating around in it. So here's a bunch of random organelles. We'll get into what some of those are in just a minute. But it has other things floating in there that are structured, that are actually organize and have specific functions, where the prokaryotic cell, it can do the same things as some of these organelles, but the pieces to it are just floating around in the cytoplasm. Basically, prokaryotic cells have everything just floating around in the cytoplasm without a whole lot of structure, whereas the eukaryotic cells have things structured in specific jobs, and so it's much more organized, and because of that, the eukaryotic cells can form more complex organisms such as animals and plants. Prokaryotic cells tend to be single-celled organisms like bacteria and other microorganisms. Now, since eukaryotic cells are a bit more complicated, I'm going to kind of go through what different parts of a eukaryotic cell. Now, I'm going to start with an animal cell here. So everything here applies to just animal cells. Start off with the things I already talked about. We've got our plasma membrane coming around there. Now, the plasma membrane on a eukaryotic cell and on prokaryotic cells will actually let nutrients go past it, so water, glucose, other things, vitamins that the cell actually needs will actually be able to pass through the plasma membrane and get into the cytoplasm. Ribosomes can use that to make proteins. It's got its nucleus with its DNA inside of it. So that's what I've already covered. Now, a bunch of organelles are also inside of the animal cell, this eukaryotic cell. So the first organelle I want to talk about, besides the ribosomes, is the rough endoplasmic reticulum. Now, the rough endoplasmic reticulum is situated outside of the nucleus. It's actually attached to the nucleus, and it forms a channel allowing things in and out of the nucleus. But it's also got a bunch of ribosomes actually studded inside of it. So it's basically just a membrane that forms a whole bunch of channels that allows things to move around as they make their way into the nucleus and out of the nucleus. It's got all these ribosomes in here. So as DNA from the nucleus makes its way out into the cell, a lot of that messenger RNA, as it comes out, genetic material, gets into this rough endoplasmic reticulum, and the ribosomes will actually start making proteins and things like that. They're embedded inside this rough endoplasmic reticulum. There are still ribosomes out here that make other proteins, but there's a whole bunch of them in here set in place, and it's easy for them to make a whole bunch of proteins because of those ribosomes. Outside of the rough endoplasmic reticulum is the smooth endoplasmic reticulum. Now, the smooth endoplasmic reticulum doesn't have any ribosomes stuck in it, which is why it's called the smooth endoplasmic reticulum. Now, this is responsible for creating a bunch of lipids. So as you get nutrients that will actually line up on the edges of this membrane here, it will create lipids, which are then moved and used for things like creating the plasma membrane and other membranes for some organelles. It has various functions. Now, as things are created in the rough endoplasmic reticulum or the smooth endoplasmic reticulum, they need to go to other parts of the cell or to other cells in a larger organism. So we have what's called the Golgi apparatus, and the Golgi apparatus actually has things move towards it, called transport vesicles, that move materials that are created to the Golgi apparatus. The Golgi apparatus actually tells those things where to go. So it's kind of like a post office for the cell. It tells it to go over to the plasma membrane or to the ribosomes, or where does it need to go to do what it needs to do. The Golgi apparatus kind of directs everything where it needs to go. Now, very important, we have these things called mitochondria. Now, the mitochondria will actually take in nutrients from around the cytoplasm, suck them in, and it will actually turn it into a usable energy source. It will basically digest it and turn it into something called ATP. Now, ATP is used by almost everything else in the entire cell, including the mitochondria, in order to fuel its functions. So it's the mitochondria that takes in nutrients and digests it down and turns it into that ATP to let other things actually use it in a very quick form. Next, we have the cleaners of the cell. We've got the lysosomes and the peroxisomes. Both of these are responsible for actually cleaning out the cell, things that might build up or things that might die. Lysosomes is actually recyclers of other parts of the cell that might die or need to be decomposed. But the lysosome will actually break it down and use that material, turn it back into nutrients that it can then put back into the cytoplasm so other things can recycle them and reuse them. Peroxisomes get their name because they're mainly used for getting rid of hydrogen peroxide, which is a byproduct of a lot of processes in the cell. And if you get too much hydrogen peroxide building up, it will actually cause damage because it's a toxin. So peroxisomes, they do other toxins as well, but they're responsible for actually collecting toxins that are in the cell and actually trying to get them out of the cell, expel them, so our body can actually expel those. Next we have vacuoles. Now vacuoles are storage areas, so it will actually store a bunch of nutrients for later use that might be needed there. And then we have the cytoskeleton. Cytoskeleton is a very rigid structure of proteins which help hold the cell together. It will actually attach itself to the plasma membrane and give it some structure, give it some support. Now, that was an animal cell. A plant cell, slightly different. We've got a couple modifications to make in order to be a plant cell. First off, the vacuoles are going to be much larger. Plant cells usually need to store a lot more materials than animal cells. Also, there's going to be a cell wall here. Cell wall is going to give it much more support, makes it very rigid. All right? And most importantly, though, we have this organelle called a mitochondria. Chloroplast. Now, a chloroplast is a special organelle which can actually turn carbon dioxide and water into glucose and store it for later energy, which is actually where it goes into the vacuole. But chloroplasts are specialized because they use the energy of the sun in order to do this process. It's the process of photosynthesis. And this is unique to plant cells. So they actually are able to make their own energy rather than absorb other organisms to get their energy. So they are a primary producer. Very important for these chloroplasts. Okay? Now, all of those are the different structures for eukaryotic cells. Getting into things that can happen to these cells, different diseases that can happen. Start off with viruses. First off, viruses, question is are they alive? Technically, no. They meet quite a few of the 8 requirements for something to be alive, but they don't quite meet them all. So let me kind of describe a virus. These are a couple of viruses. We got the HIV virus, the rabies virus, the influenza virus, but viruses cause a lot of damage to cells. So what they actually consist of, all viruses have the exact same, have a couple of common parts. One is a capsid. Now this is not a plasma membrane. It's a protein coat rather than a lipid coat. So, but it's similar function to a plasma membrane, and it encapsulates the virus. Now, the virus also will have genetic material. All viruses have these 2 common things. They might have more, but they have at least these 2 common things. Genetic material, which is DNA or RNA, and a capsid to encase it. They don't have much else, though. So they're very, very, very simple things. Now, sometimes the viruses will have an envelope, which is some kind of plasma membrane, which is actually what you're seeing right here. It will have little things sticking off of it, which help it to attach to things, different cells. Maybe it'll have other things attached to that envelope, but depends on the virus. All right. They definitely have the capsid and the genetic material, though. So here's the way a virus works. This is a cell. It's a eukaryotic cell. The virus will actually attach to it, and depending on its envelope, or whatever it has sticking outside of it, will attract it to different types of cells. So this one is attracted to this type of cell right here, so it attaches to it. Now after it attaches to it, it'll actually inject its genetic material into the cell. All right, and that genetic material will make its way into the nucleus of the cell, and it'll actually replace some of the genetic material of the cell. So it'll insinuate itself into the genetic material of the actual cell. Now once it's in there, the cell does its thing and it tries to use that genetic material to make other parts. That genetic material is being used in other parts of the cells to create things, but instead of creating the things that the cell needs, it will create things that the virus needs. It'll actually start creating parts for new viruses. And those parts are being created by the ribosomes and the other parts of the cell. Once they're created, they start assembling themselves into new viruses, and eventually there's so many new viruses in the cell that the cell will actually burst and they'll break out. You'll have tons of new viruses go everywhere to other cells, and the whole process starts over again. Right? Where the virus attaches, inserts its genetic material, creates new viruses that burst out and finds new cells. It just keeps growing and growing. So this does in fact meet some of the requirements for life, where in the fact that it actually has genetic material, it grows, things like that. But this is actually not technically a cell. Also, it cannot reproduce without help from another organism. So it cannot reproduce on its own. So this is too simple to actually be considered a life. It doesn't meet all the criteria, but it is very, very close. It's one of the closest things we found. Now, a prion. Prion is a type of disease which also is not actually alive. It's actually part of a living organism. So in order to describe how prions work, first we have to talk about proteins. So we have these proteins, and this is actually drawing proteins in the brain, because usually this is a brain disorder. So proteins are folded a specific way, and these proteins in the brain are used by the cells of the brain to do a specific task, and it needs those proteins to do that task. Now, a prion is the exact same protein as these. Prions are those proteins, except it is folded incorrectly. Right? So it's a slightly different shape than what it's supposed to be. Right? And because of that, it doesn't actually perform its function correctly. Now, the thing with a prion protein, though, because it is folded incorrectly, if this prion protein comes across a normal healthy protein, it will actually cause it to unfold and refold into the unhealthy form. So if you get one prion which is unhealthy and folded incorrectly, it will actually cause another protein to unfold and refold into an incorrect form, turning it into a prion as well. And then that one comes across some of the other healthy proteins and turns them into prions. And then those turn the rest of them into prions, and this is a cascade effect. It keeps getting faster and faster. The more prions you have, the more proteins get turned into prions. And it just keeps going, and it degenerates, and eventually it causes a severe mental disorder, because then you don't have the correct protein doing its job in the brain. So this causes diseases such as mad cow disease. So it's not technically alive because it's actually a part of the cell, but it is a very debilitating disease. Now, what does all this have to do with what I was talking about in the beginning with finding new Earth-like planets? Well, we have to understand exactly what life is if we want to actually find life on other planets. So as we're looking across the stars and we look at other planets, look at these 7 things, we're going to be looking for some of these characteristics. We're going to be looking for signs of cells. We're going to be looking for signs of responses to the environment, things that can't happen without organisms. So we'll be looking for things like oxygen, because lots of organisms will give off oxygen, or we're looking for carbon dioxide, or we're looking for water. All of these things are things that cells and life actually need. If we actually were able to get there, first thing we're going to be looking for is cells. We're going to be looking for microorganisms. So we have to be able to identify what a cell looks like. So that's my presentation on cells. Sources here for the information in this presentation: Hidden Kingdom, part of the BBC program, the Cell series, which was published in 2009. I also referenced Paul Anderson, part of Bozeman Science, a YouTube series, specifically his series on viruses, which was published in 2012, and his series on prions. And then we had Biology for Dummies, the 2nd edition, which was written by Reen Fester-Kratz, PhD, and Donna Ray Siegfried. That was published by John Wiley and Sun Incorporated in 2010. Also, I referenced NASA's website, which you can go to at nasa.gov. Thank you, and I hope you enjoyed this presentation.